We're going to take a look at the spinal cord in three different ways. We're going to think about firstly the spinal cord segments, then we're going to take a look at the grey matter of the spinal cord, and we're going to finish off by looking at the white matter of the spinal cord. Now, the spinal cord is organised in separate segments which reflects the segmental anatomy of the human embryo. You'll recall back from when you studied embryology in detail in the first year that we had a neural tube and we had somites, for example. The neural tube went on to form the brain and spinal cord, and the somites went on to form muscles, contributed to the skin, and also helped to form the vertebrae themselves. And there is a one-to-one correspondence between spinal cord segments and the derivatives of the somites. Now, there are around 31 spinal cord segments. I say around 31 because the coccygeal segments are variable in number. And each spinal cord segment has a left and a right half, which in turn supplies a given dermatome and myotome on each side of the body. If you're a little bit hazy on the definitions of dermatome and myotome, I do recommend that you go back to your notes from MSK. Now, if we take a little bit of a closer look at the spinal cord anatomy, we can see here is the spinal cord, this cylindrical structure. We can see it is definitively split into left and right halves by these very clear fissures at the front and back of the spinal cord. And we can see that we have elements of the peripheral nervous system here in yellow plugging into the cord itself. The basic anatomy of a spinal cord segment is that it has this H or butterfly shaped central core of grey matter surrounded by an outermost layer of white matter. And in fact, this is the fundamental pattern of the entire central nervous system, really. It just so happens that within the cerebral hemispheres there is an additional superficial layer of grey matter added on, which is the cerebral cortex. Now, each of these spinal cord segments on the left and right sides has two structures plugging in. It has the so-called dorsal root and the ventral root. The dorsal root is at the back and the ventral root is at the front. However, you can see really we're not talking about a single root going into each spinal cord segment, but multiple rootlets. So there are a variety, there's something like 5 or so distinct rootlets joining into each cord segment at the back to form the dorsal root and at the front to form the ventral root. So the roots are formed from the convergence of the rootlets. Now, the dorsal root is sensory. Okay, the dorsal root is purely sensory, whereas the ventral root is purely motor. And when I say purely motor, I mean that it includes somatic motor fibers going to the skeletal muscles, but in addition it will also contain autonomic fibers depending upon the level within the nervous system that the ventral root is found at. You can furthermore see that the dorsal root has a swelling known as the spinal or dorsal root ganglion, and this is where the cell bodies of first order sensory neurons are found. We then see that the dorsal and ventral roots coalesce to form the spinal nerve, which very rapidly splits to form the most important ventral ramus, which goes on to contribute to plexi such as the brachial and lumbosacral plexi. But additionally, the spinal nerve splits into the dorsal ramus, which supplies the epaxial muscles of the back, i.e. the intrinsic muscles of the back, such as erector spinae. Now, by knowing that the dorsal root is purely sensory and the ventral root is purely motor, and that they form a mixed spinal nerve, you already have some very powerful pieces of knowledge which allow you to localize lesions within the nervous system. For example, at the most basic level, if you saw a patient who had an isolated sensory loss within a single dermatome with no associated motor features, you could localize quite confidently to the dorsal root. Whereas if they had weakness within a given myotome with no associated sensory features, you could localize to the ventral root. If a patient had mixed motor and sensory features within a given dermatome and myotome, you might localize to the spinal nerve. So really, this dermatomal and myotomal supply is incredibly important to enable you to localize lesions within the nervous system. Before we move on to our next portion, just remember the fact that the vertebral column grows faster than the spinal cord. This is very, very important and has important clinical consequences. What it means is that at around about the cervical and upper thoracic levels, there's pretty much a one-to-one correspondence between vertebrae and spinal cord segments. However, once we get down to the lower regions, we can see that the roots, the dorsal and ventral roots, have been pulled out, drawn out by the rapid growth of the vertebral column, leaving this structure known as the chorda equina that fills up much of the vertebral canal at the lumbar level. By knowing about this, we know that we should, in theory, be able to safely perform a lumbar puncture, say at this level here, without risking damage to the delicate spinal cord, which has petered out at around about the L1 or L2 vertebral levels. So there's some very important anatomy here. Let's move on. Next, we're going to think about the white matter of the spinal cord. Now, we said that the spinal cord contains a central H or butterfly-shaped region of grey matter surrounded by white matter. Okay, and it is this white matter that we're going to focus on. Within this white matter, we have the major pathways enabling the brain to communicate with the rest of the body via the spinal cord. We have descending motor pathways going from the brain down to the cord, and ascending sensory pathways going from the cord up to the brain. And these pathways can be split anatomically into three distinct classes. The largest subdivision of the spinal cord white matter is known as the funiculus, plural being funiculi. And a funiculus we can define as a segment of white matter containing multiple distinct tracts. Those impulses travelling within a given funiculus move in multiple directions. That is, they might move up or down within those various distinct tracts within that given funiculus. So looking at the spinal cord, we have the dorsal funiculus at the back, the lateral funiculi at the sides, and the ventral funiculi at the front. And strictly speaking, we have a funiculus on each side. So we have got a left dorsal funiculus, a left lateral funiculus, and a left ventral funiculus. Now within the funiculi, we have structures known as tracts. And the tracts we can see quite nicely shown on this figure on the left hand side. We have ascending tracts in blue and descending tracts in green. Now a tract is an anatomically and functionally defined white matter pathway which connects two distinct regions of grey matter. And importantly, in a tract, impulses travel in only one direction. So tracts may ascend, and they are sensory, or they may descend, which makes them motor. Finally, we can take a tract and we can actually split it into smaller subdivisions, and we can call these subdivisions fasciculi. And a fasciculus we can think of as a subdivision of a tract, which supplies a distinct region of the body. Now, in later lectures, we'll study the major pathway within the dorsal funiculus, which is known as the dorsal column tract, and that tract is split into two fasciculi, the fasciculus cuneatus sitting laterally and the fasciculus gracilis sitting medially. The gracile fasciculus sitting medially supplies the lower half of the body, whereas the cuneate fasciculus sitting laterally supplies the upper half of the body. So a fasciculus is a subdivision of a tract, usually supplying a distinct region of the body. So let's move on to our final portion, where we're going to look at the grey matter of the spinal cord. Now, it is much more important for you to know about the white matter of the cord than it is to know about the grey matter of the cord. However, you still need an appreciation of what's going on here. Now, just as we were able to subdivide the white matter of the cord into distinct tracts and fasciculae, etc., we can subdivide the grey matter of the cord. But the grey matter of the cord isn't divided into pathways as such, it's divided into cell columns. Now, why am I talking about the idea of cell columns? Well, if you take any distinct area of grey matter in the cord, you've got to remember, so for example the ventral horn here of the spinal cord, you've got to remember that this is a three-dimensional structure. So it's almost as if we'd be looking at a cross-section through a sausage roll here. But we have to remember that the sausage meat in the middle is a three-dimensional cylinder running through the cord itself. So really we can think about the grey matter of the spinal cord as being made up of cell columns, cylinders of cells running longitudinally through the cord. Now, these cell columns have been studied in detail, and the guy who looked at these most closely was a guy called Rexed, and he gave these cell columns particular numbers, and they became known as Rexed's laminae. And you don't need to know these, but I'm presenting them to you just so you can see the rationale for what Rexed did. What Rexed was able to do was to look microscopically at the grey matter of the spinal cord and to identify that there were histological differences between the organisation of the cells within these different regions. And because function follows from structure, we then learnt that these different Rexed's laminae corresponded to different given functions. For example, we discovered that lamina 9, represented by these two columns here, contained the motor neurons that went on to supply muscles. So you don't need to know Rexed's laminae, but what it does tell us is that there are specific three-dimensional columns of cells within the cord, which gives it a subdivided appearance. Now, one final point before we leave the spinal cord grey matter is just to think about a myotome. You should remember that a myotome is the muscle or regions of a muscle supplied by a given spinal cord segment. Actually, when we look at this area in a bit more detail, we discover that each individual muscle is in fact supplied by at least two distinct myotomes. But taken as an average, most muscles receive innervation from just two cord segments. If we look at the figure on the right hand side here, we can see, for example, that if we take iliopsoas here, these are all lower limb muscles. If we take iliopsoas, we can see that iliopsoas is primarily supplied by the L1 and L2 spinal cord segments, i.e. iliopsoas is supplied by the L1 and L2 myotomes primarily. So what you've got to imagine is that within the L1 and L2 cord segments, we have a three-dimensional cylinder of cells, which are the cells responsible for activating iliopsoas. Looking a bit further down, we can see that gluteus medius and minimus are primarily supplied by the L5 and S1 myotomes. So if we had localized damage to just a single region of the spinal cord, it may be the case that a muscle is not completely paralyzed due to the contribution from the adjacent myotome.